To illustrate this, we will use a simple model of a muscle fascicle that consists of only 6 muscle fibers innervated by 4 motor neurons. In addition to its motor neuron, each fiber has an arteriole that supplies it with blood for the oxygen and nutrients needed for energy essential to contraction. Finally, the fascicle is innervated by pain-sensing neurons called nociceptors that are sensitive to the absence of oxygen and the presence of ATP and inflammatory chemicals. The formation of a fiber trigger point starts with its motor neuron. Each fiber in a muscle is innervated by a single motor neuron that controls the fiber contraction. Nerve impulses in the motor neuron are conveyed to the fiber surface from the neuron's motor end plate. The end plate contacts the fiber in only a single location. The electrical impulse passes from the end plate over the fiber surface and causes the fiber to undergo a full cycle of contraction-relaxation. Here is a close-up of a motor end plate. This view is simplified to show how the end plate stimulates the fiber contraction. In a previous scene, a simple model was used to describe how acetylcholine is released from the end plate, binds to an acetylcholine receptor to open a sodium channel, and produces an electrical impulse that spreads over the fiber surface and causes a contraction. To explain trigger point formation, the process is actually somewhat more complex. In reality, the fiber surface beneath the end plate has many receptor channels. Acetylcholine is actually released in units of around 10,000 molecules. Each acetylcholine unit activates a region of the fiber surface and produces a localized depolarization in that region called a miniature end plate potential. When motor neuron action potentials reach the motor end plate, the entire end plate is activated and 1 to 2 million acetylcholine molecules are released all at once. This mass release of acetylcholine produces widespread synchronous miniature end plate potentials that fuse into a grand end plate potential and produce a fiber depolarization that is then propagated over the length of the fiber surface, resulting in fiber contraction. However, acetylcholine can be released in lesser amounts than a maximum release. End plates can spontaneously leak acetylcholine molecules, and individual acetylcholine receptor channels are activated and form local depolarizations that are detected as spontaneous background end plate activity. It is also normal for motor end plates to release entire units of acetylcholine spontaneously and generate individual miniature end plate potentials. These individual miniature end plate potentials are too uncoordinated to form the grand end plate potential needed for a fiber contraction. However, when a muscle is damaged by trauma, exposed to infections, or undergoes prolonged tension by repetitive or persistent contraction, it can become inflamed, and the motor end plates become hyperirritable. Hyperirritable end plates show recurrent bursts of acetylcholine units that produce numerous uncoordinated miniature end plate potentials over the surface of the muscle fiber immediately below the end plate. Although abundant, these miniature end plate potentials are erratic and scattered and remain below the synchronized depolarization needed for a full end plate potential, and therefore fail to generate an action potential and a normal cycle of full contraction relaxation in the affected fiber. Nonetheless, the recurrent miniature end plate potentials appear to activate that sequence of molecular processes that cause a fiber contraction, but only in those sarcomeres that are immediately under the motor end plate. Since hyperirritability and its bursts of acetylcholine and the generation of miniature end plate potentials are all ongoing, the affected sarcomeres eventually undergo a maximum shortening and remain locked in a supercontracted state. The persistent unsynchronized bursts of neurotransmitter and their resulting weak but constant miniature end plate potentials result in a series of sarcomeres that accumulate into a hypercontracted knot in the length of the fiber under the end plate. Let us see this from the perspective of an intact muscle fascicle. Based upon our previous close-up, the fiber surface is being exposed to uncoordinated bursts of acetylcholine by the hyperactive end plate, and in response, it is generating multiple miniature end plate potentials. This causes the sarcomeres under the end plate to undergo maximal shortening, but with no relaxation. As the initial sarcomeres contract, neighboring sarcomeres are dragged under the end plate and stimulated to contract. Sarcomeres do not change their volume, so that sarcomeres under the end plate that are stimulated to shorten become wider, but adjacent sarcomeres outside the end plate that are not stimulated to shorten become stretched and thinner. This results in a knot forming in the fiber in the region under the end plate. Let us now explain how the miniature end plate potentials cause sarcomere hypercontraction. This knot is the trigger point and has profound disrupting effects on the physiology of the muscle fascicle. Formation of the knot compresses the arterioles of the affected fiber and restricts blood flow, which is one of the main causes of trigger point formation at the level of the sarcomere. The reduced blood flow diminishes energy production in the hypercontracted fiber, and since ATP is necessary for both the contraction and relaxation phases of the cycle, once the ATP has been consumed by the contraction phase, the contracted sarcomeres cannot return to their relaxed state. Let us take a closer look at the start of trigger point formation to better understand how trigger points form and why they cause pain. Let us look inside the fiber at the sarcomeres to see how they respond to the constant release of acetylcholine by the dysfunctional end plate. Here are a few of the key structures inside a fiber. The fiber is the cell of the muscle, so we are looking at the structures and their organization inside a muscle cell. Of note are the long myofibrils that are comprised of sarcomeres, which contain the contractile elements of actin and myosin. Filling the inside of the fiber is a fluid-like cytoplasm called sarcoplasm, which is not shown. Also scattered around between the myofibrils are mitochondria that synthesize ATP, which serves as the chemical energy to power muscle contraction and relaxation. We have already discussed the role of the sarcoplasmic reticulum to store calcium, and the T-tubules that transmit the electrical impulse from the outer fiber membrane into the fiber and over the surface of the sarcoplasmic reticulum to cause calcium release and initiate contraction. A myofibril will be magnified to better observe the effect of miniature end plate potentials on the sarcomeres during trigger point formation. Although miniature end plate potentials are inadequate to stimulate a full fiber contraction, they are nonetheless able to spread over the fiber membrane and into the T-tubule system and cause release of small amounts of calcium from the sarcoplasmic reticulum. The released calcium is sufficient to activate the actin-myosin cross-linking and produce contractions by just those sarcomeres under the active end plate. Sarcomeres cannot alter their volume, and shortening during contraction causes them to widen. As the sarcomeres contract and widen, the arterioles that carry glucose and oxygen to the fiber become compressed. Sarcomeres require ATP for both contraction and relaxation. Adenosine triphosphate, or ATP, is the main form in which energy is transferred and stored within cells. ATP is synthesized by mitochondria as part of the oxidative metabolism of glucose. The terminal phosphate bond of ATP is a high-energy bond that can release its energy to drive cellular processes. Normal cells are replete with ATP. Here we see the contraction-relaxation cycle of actin and myosin and the critical role for ATP in this cycle. During the contraction-relaxation cycle, the myosin head is either bound to ATP or to actin. Myosin is bound to ATP in preparation for cross-linking and only releases ATP as it binds to actin. Likewise, myosin remains bound to actin until it can again bind ATP. Only then does it release from actin. During hypercontraction, blood flow is restricted by the swelling sarcomeres, and the increasing ischemia suppresses ATP synthesis. Decreasing fiber ATP prevents bound myosin head units from releasing actin, and the sarcomere is held in a progressively contracting state. As the sarcomere reaches its maximum contraction during trigger point formation, it is being continuously stimulated to contract by the constant barrage of miniature end plate potentials causing calcium release. This causes the sarcomere to become hypercontracted. As the first sarcomeres shorten, they draw adjoining sarcomeres under the end plate that also initiate contraction. Increased energy demand at the same time as metabolic supply is impaired results in a localized energy crisis. Without further ATP synthesis, myosin cannot release from its actin crosslink. The calcium pump is also sensitive to low ATP and cannot return calcium to storage in the sarcoplasmic reticulum so that the contractile mechanisms continue to be stimulated. Other hypercontracted sarcomeres accumulate into the knot under the dysfunctional end plate. The stressed sarcomeres are also damaged and the fiber releases cellular contents into the fascicle. Local ischemia, combined with the release by damaged fibers of inflammatory chemicals such as bradykinin, substance P, and histamine, stimulates the fascicle nociceptors to generate pain signals. We will now look at the relationships between muscle trigger points and pain.
Musculoskeletal pain. Myofascial trigger points are a common cause of non-articular musculoskeletal pain. Myofascial trigger points are discrete, palpable, hyperirritable nodules in a taut band of skeletal muscle that can only be diagnosed by systematic palpation of the soft tissue by an experienced examiner. Activation of group 3 and 4 nociceptive afferents causes the pain and tenderness experienced at myofascial trigger points. One mechanism of musculoskeletal pain is initiated when noxious stimuli deform the axonal membrane of muscle nociceptor endings beyond their firing threshold, causing the release of a variety of biochemicals such as bradykinin, prostaglandins, and serotonin from muscle tissue, which may stimulate group 3 and 4 muscle fibers. This biochemical cascade can also be elicited without mechanical trauma to the muscle. Once sensitized, group 4 afferents fire at lower thresholds, responding to innocuous stimuli like weak pressure and muscle movement, causing the exquisite tenderness of active trigger points. This phenomenon is called peripheral sensitization. Continuous input from myofascial trigger points can increase excitability of dorsal horn neurons. This phenomenon is called central sensitization. This can cause allodynia and hyperalgesia and open previously ineffective synapses, resulting in new receptive fields and the referral of pain. Stored substances in nociceptor endings also affect local microcirculation through vasodilation and plasma extravasation. Nociceptive input from skeletal muscle is much more effective at inducing neuroplastic changes in the spinal cord than input from the skin. This dynamic process can fundamentally alter pain threshold, pain intensity, and emotional affect.